So this is work of ours from Naples. My name's Brian Dale. We set up this laboratory in Naples about 20 years ago. And one of the things we've been interested in is age and reproductive success. So if we look, so the objectives of this talk: what is the maternal age effect? We all know about it. Do we understand the metabolism of oocytes and embryos? Do we really understand that? Our hypothesis of maternal age with respect to metabolism. And can we change things? Can we improve embryos by doing something to the metabolic status of the embryo? So what is the maternal age effect? We've known from years that with age it's more difficult to reproduce. The first observation really was, I suppose, these observations where you see that you get an increase in trisomy with age. So if you see a woman of, say, 30 years of age having 0.4, when we go to 50 years of age, we're talking of 40, so 100 times more trisomy in a woman of an age 20 years older. Now, more often we would think of, at least in the context of reproductive biology, that the pregnancy rate goes down. So from a woman of 30 years of age to a woman of 40 years of age, it shoots down, and then afterwards it's even more difficult. We know it's an oocyte problem. It's not the uterus. And we know that in the oocyte there are increases in chromosomal problems. But what are the mechanisms? So chromosomes, yes, you have aneuploidy, it's wrong, it's very gross. It's not a genetic thing. I mean, so you've got an extra chromosome or less chromosomes, you get a big problem. But is that really the mechanism? Or what is causing those chromosomal mechanisms? So why is there one extra chromosome or one less chromosome? Whatever it is, the maternal age effect, it could be to do with protein synthesis, mistakes in DNA synthesis, structural problems of creating a meiotic apparatus. But anyway, all of these processes, whatever you do in a cell, you need energy. There's two pathways basically in cells, anaerobic and aerobic. Aerobic's much more efficient, and in most cells or embryos you have both these pathways functioning. For example, in the human we have 56% goes through an aerobic respiration pathway. Obviously this data needs a lot more work on it to understand exactly what's happening. But so what are mitochondria? Mitochondria you find in most cells, most eukaryotes. In the oocytes, mitochondria are derived from a few mitochondria that you find in the small primordial oocyte, and then are replicated as the oocyte's growing. Oocytes contain 100 to 600,000 DNA copies in the mitochondrial setup, and we know that from studies that if there are less mitochondria in an oocyte, the person is less fertile, less probable to have a pregnancy. The number of mitochondria we have in the oocyte are the numbers that we have in the first 5 days of development. So that it's obviously important that we have a good number of mitochondria in the oocyte from the beginning. In the human oocyte, if you measure mitochondrial activity, it's actually quite low. And a good component is actually from aerobic mitochondrial respiration in the oocyte. Okay, so you can actually measure ATP, and the average amount of ATP is 1.85 picomoles per oocyte. Van Vlerken years ago showed that if you have more than 2 picomoles, then you've got a potentially good oocyte. Mitochondrial respiration, we know from other systems, is correlated with aging. Mitochondrial DNA code for proteins involved in respiration. There's little protection for mitochondrial DNA, so if they have problems because of free radicals or whatever, they're soon messed up mitochondrial DNA. For example, free radicals cause a lot of problems in the oocyte, and as we know in human reproduction, if you look at slow dividing cells like neurons, there's very low aerobic respiration. And the primordial oocytes, we know, are very prone to free radical attack. Okay, so what do we have in the human? We have the fact that you've got oocytes in the human body for 40 years, exposed to all sorts of things as the woman is in the life of the woman. Mitochondria, you could look at the genetics of mitochondria. We're not interested in that. We're physiologists, and so we're looking at the actual live activity of mitochondria. And you can do that filling the oocyte or a cell with a dye, and then put the oocyte under a confocal microscope and look at the fluorescence emitted from that dye. And we started working on mitochondria in the human system from 2001. Okay, this is all genetic stuff. And we found, measuring from over 80 M2 oocytes, that with age, mitochondrial activity, now we're not interested in genetics at this moment, decreases. A very, very clear correlation. So oocytes from an older lady, even though they're M2, have less mitochondrial activity, which isn't surprising. So these are live confocal images. So here, in this particular case, we're looking at two apparently normal M2 oocytes, human oocytes. If you fill them with a dye, and you look at mitochondrial activity but also distribution, you can see in this typical situation, and then if you label later for the meiotic spindle, you've actually got a very normal spindle with a good normal attachment of chromosomes in red. This also looks very much like this oocyte. It's actually from an older lady. Mitochondria generally is less. There is a distribution problem, but it's actually, in this particular case, it's actually, we're looking at the general activity, and you can see that the meiotic fuse is messed up. That was a general trend we found in many oocytes. If you look at embryos, same thing. So here we're interested in, these are all apparently good 4-cell embryos. But if you look at the mitochondrial activity, here they're all much the same, whether there's mitotic disjunction, normal, meiotic non-disjunction. Only in the case of chaotic mosaics, where the whole embryo is messed up chromosomically, it's a real mess, can you find a correlation with mitochondrial activity? So what does the data suggest? So meiotic non-disjunction, which is very common in the human, is not correlated with low mitochondrial activity. Chaotic mosaicism, where the whole embryo is all messed up, each embryo, each cell is different from the other one, is correlated. This again was from our work in 2003. So what's our hypothesis about this? So as maternal age increases, as the woman gets older, you get low developmental potential. So the women are less likely to have a baby, lower pregnancy rates. So our hypothesis is that the mitochondrial activity in the oocytes of the older lady is reduced, and this is having, as one of the effects is on the chromosomes. We're not really very interested in chromosomes. As I say, it's a very gross change to have one or less chromosomes. But generally we think that mitochondrial activity programs the developmental fate of the human pre-implantation embryo. If mitochondria are so important, and we're in the laboratory and we have oocytes and embryos in the laboratory, can we change things? Dr. Tucker talked about medium and the importance of medium. Can we do other things? The nicest technique, but unfortunately it's been banned throughout the world, is cytoplasmic transfer. So what does that mean? So Jack Cohen, a friend of ours, all of ours, did this. He took oocyte, cytoplasm from an oocyte of a young patient and put it into the cytoplasm of the other patient. And the idea generally was to increase the level of healthy mitochondria or to block degenerative processes. So we're improving the cytoplasm of the oocyte. We're making a better environment in the cytoplasm where obviously the chromosomes are trying to work, and that could also reduce aneuploidy, if aneuploidy is part of the problem of... Jacques had a very high success rate, 35%, in doing cytoplasmic transfer. Groups in Israel did it, we did it quite a bit, we've published, we've got pregnancies, but then of course the various governments didn't like it, they think it's invasive, and it was stopped. Another mechanism would be to do general vesicle transfer, fuse a GV with a cytoplasm and mature oocyte, so fuse two cells together basically, reconstruct a cell, and then fertilize the cell, and that's another way of improving the cytoplasm. It's on my talk, you can see that. So I'm struggling with this because I didn't do it. So mitochondrial activity decreases with maternal age. That's a fact. We've measured it, we show it. In the short term, mitochondrial ATP production can be compensated because the cell can go the other way and do anaerobic, the anaerobic pathways through the cytoplasm. But it's not sustainable. You need mitochondria. It's going to burn out. You need both ways, even though they depend on each other. So, as I said, compensation cannot go on for very long using the anaerobic pathway. Techniques are under development. We think people should be doing cytoplasmic transfer everywhere in the world, and we should be doing these types of things. The problem in Italy, for example, is you have to use cytoplasm from a donated oocyte. Italian government doesn't like the fact we donate oocytes, but maybe there should be ways around it saying, well, it's a bit of cytoplasm, it's not an oocyte. Last thing I'd like to say, it was the latest work that Martin put together, which was an epidemiological study on the Scandinavian population, and this sounds a bit frightening at the moment, but it's a fact. He looked at 18,000, what do you call it? He looked at the anographic data in Sweden, I think it was, 18,000 people, and correlated, and it was negative correlation, with age of your mother at birth with death of that person, and found out, which is not very nice, that if you were born from a woman of 40 years of age rather than a woman of 20 years of age, your mother, you're more likely to die by about 10 months. Your lifespan is shorter by 10 months, excluding all diseases, cancers and everything else. So basically, taking all away pathologies, the answer to that would be have a baby at 20 years of age. Unfortunately we don't, we'd have it at 40 years of age. So we need to look at mechanisms to improve oocytes from women of 40 years of age. Thank you.